All right, for Chapter 2, Chemistry of Life. This chapter will have a good deal of detail. Hopefully it will mostly be a review with just a little bit of new material. There are several videos throughout this presentation, all of which have been linked in Blackboard under Chapters 1 and 2 videos. Also be sure to read Chapter 2 section objectives before you read each section. Although this is a biology class, much of what happens in living organisms is driven by chemical processes and interactions between atoms, molecules, and elements, so a little refresher on basic chemical ideas is important to understand much of what we will discuss later this summer. You probably remember that matter is anything that has mass and takes up space, but what about an element? An element is a substance that cannot be broken down into a more simple substance. All known elements are found on the periodic table of elements. A few elements you may be familiar with would be helium, aluminum, gold, silver, or possibly neon. In living organisms, there are 6 elements that are considered the building blocks of all other chemicals in an organism. They are sulfur, phosphorus, oxygen, nitrogen, carbon, and hydrogen. If you take the first letters of these elements, you can spell the word sponge to help you remember. You will hear much more about carbon and hydrogen as they are found more than the rest in the living organisms. Atoms are particles which combine to make an element. Each carbon atom will have properties like all other carbon atoms, but will be different from all, let's say, oxygen atoms. Within an atom, there are 3 smaller particles that make each atom of an element unique from atoms of other elements. An atom has a nucleus where 2 of these subatomic particles are housed. Protons, which carry a positive charge and have mass. The number of protons helps identify the atom. Therefore, for instance, an atom with 6 protons would be identified as carbon, while an atom with 8 protons is known to be oxygen. Neutrons are also found in the nucleus, and they also have mass, but they do not have a charge like the proton. And then outside of the nucleus, in an area known as the electron cloud, electrons zip around the nucleus. These particles have almost no mass, but they carry a negative charge. The interaction between positive protons and negative electrons is very important in living organisms. A molecule is when 2 or more atoms are chemically bonded. Water is a molecule since it has 2 hydrogen atoms bonded to an oxygen atom. And even 2 oxygen atoms bonded to each other is a molecule of oxygen. And 1 carbon bonded to 4 hydrogens is a molecule of methane. 2 terms to keep clear. The atomic number is the number of protons in an atom's nucleus. This was the one used to identify an atom. The atomic mass, also referred to as mass number, is the mass in the nucleus. The only 2 particles in an atom that have any mass, protons and neutrons, are both found in the nucleus, so all the mass is found in the nucleus. And to add one more term to the atom, we mentioned electrons move around the atom outside of the nucleus. There are different levels or shells where they move about. The level farthest from the nucleus is where electrons come and go from an atom. These electrons are known as valence electrons. They are very important in understanding how chemical reactions happen between atoms and molecules in organisms. There's something called the octet rule that refers to the concept that an atom prefers to have a full valence shell, and they like to have 8 in that last shell, except for hydrogen and helium. They only like to have 2. So these atoms will gain or lose electrons in order to satisfy that octet rule. In the bottom picture, it represents how the red electron is moving from the sodium, or Na, with the purple nucleus to the chlorine, or Cl atom, with the green nucleus. Valence electrons move to fill or empty a level. the sodium atom started with one lonely electron in the valence shell, so it found an atom of chlorine that had 7 electrons and therefore room for one more in its last shell. The bottom two atoms show what sodium looks like without the red electron and chlorine looks like with the red electron. Now both of these have valence shells. The last shell that has any electrons in it, they're both full with 8 electrons. A few more important terms just that you'll hear throughout the summer. Isotopes are different forms of the same element. This means they will have the same number of protons. To retain the properties of the element, they have to have the same number of protons. But the number of neutrons is different. And if you remember, neutrons have mass. So when neutrons change, the mass of the atom will also change. The image of the two oxygen isotopes show that they both have 8 protons, but the atom on the left has 2 additional neutrons, which makes it slightly heavier than the other atom of oxygen. Scientists know the rate at which these atoms change and can therefore use them for dating fossils, or we also can use carbon for this as well. So carbon dating is that same idea. Ions are atoms where electrons and protons are not equal, resulting in an overall charge of an atom. An atom with more protons than electrons will have an overall positive charge and are known as cations. When an atom has more electrons than protons, the overall charge is negative, and those are called anions. On the fluorine diagram, notice on the left there are 9 negative electrons and 9 positive protons. These particles cancel each other out and leave the atom with an overall neutral charge. In the atom on the right, it has gained 1 electron, which is negative, so now there are 10 negatives to 9 positives. This would leave the atom with an overall charge of -1. The fluorine atom has become a fluorine ion. When atoms connect, they are said to have bonded. These bonds hold atoms together to form molecules, and there are many molecules which are crucial to making a living organism function. Ions, which remember are atoms with unequal electrons and protons, are attracted to opposite charges and therefore create a bond. For example, when sodium gives up its electron, the red dot, and chlorine accepts it, it leaves sodium with 1 more proton than electron and therefore an overall positive charge, creating a cation, and chlorine now has 1 more electron than proton, so its overall charge is negative, creating an anion. As a result, these two ions are attracted to each other and form a bond, an ionic bond. Covalent bonds, on the other hand, are when 2 atoms both make use of a few of the same electrons. They share those electrons to fill the octet rule and fill the valence shells. Looking at these 2 chlorine atoms, they each have 7 electrons, but if they bond, they can each benefit from each other filling the valence shell. When 2 atoms share electrons, they can form one of 2 types of covalent bonds. Polar bonds are drawn to one atom's nucleus more than the other. They are pulled to one pole, if you will, of the molecule more than the other. This is the case with water molecules. The electrons are more attracted to one side of the molecule than the other, and is therefore considered a polar molecule. Atoms which form nonpolar bonds spread out more evenly. In the case of CH4, all 4 of the hydrogen atoms are spread evenly around the carbon. There are no pulls to one side or the other, and therefore it is considered nonpolar. 2 other types of bonds or attractions between atoms are hydrogen bonds and van der Waals. Hydrogen bonds are very weak and are easily broken, but are very important to sustaining life. Van der Waals interactions are also weak attractions that occur between covalent polar bonds of different molecules. As mentioned previously, water is a polar molecule with one side that is slightly positive and one side that is slightly negative. This slight charge on the atoms creates the unique shape of water as hydrogens repel each other. When other substances are easily dissolved in water, they are called hydrophilic or water-loving, while substances that do not mix well with water are referred to as hydrophobic or water-fearing. Water is able to release and absorb heat more slowly than many substances. Water also has several important properties which makes it important to life. It is very important as a solvent, and its cohesive and adhesive characteristics help attract it to water molecules as well as other surfaces. Cohesion is when molecules stick to themselves, while adhesion is when a molecule can stick to something else. If I were to say I will adhere this paper to the refrigerator, it means I'm going to stick the paper to the fridge. If you've ever heard the terms acid or base, then you were probably discussing the pH of a substance. pH refers to how many hydrogen atoms are present. The scale in which this is measured is referred to as the pH scale. Low numbers are acidic and have more hydrogen ions, while numbers higher on the scale are basic or alkaline and have fewer hydrogen ions. Buffers exist to help regulate pH and keep it where it should be. Carbon is very unique in that it can form 4 bonds and is therefore able to bond with more elements than any other type of atom. It can also form 3D shapes. Any of these bonds can be single, double, or triple. Notice in the diagram, the top image, A, shows a very long string of carbons bonded. B shows a double bond with elements including nitrogen, while C shows a ring 3D shape, just to show how versatile carbon bonding can be. There are 4 macromolecules necessary for life: carbohydrates, fats, proteins, and nucleic acids. Carbohydrates have a ratio of 1 carbon to 2 hydrogen to 1 oxygen, but are usually much bigger molecules like glucose or table sugar, which is C6H12O6, still the same ratio. They can be single sugars called monosaccharides, as seen at the top of the image. 2 sugar units are called disaccharides, as indicated in the middle of the image, or a string of many sugar units would be known as a polysaccharide. Carbohydrates are typically recognized by the OSE at the end of their name, such as glucose, fructose, maltose. Monosaccharides are the simplest of the carbohydrates. They are usually 3 to 6 carbons long, as noted in the image. These 3 examples are all hexose sugars since they are 6 carbons long. Glucose, galactose, and fructose are isomers. They are all the same formula, but with different arrangements, and when the arrangement changes, their properties change. So notice how all 3 of the sugars in the image have the same number of carbons, hydrogen, and hydroxides are OHs, but they're arranged differently. Disaccharides are 2 simple sugars which have been bonded through a dehydration process. After they bond, a water molecule is released. Some common disaccharides are sucrose, which is 1 glucose and 1 fructose, lactose, which is milk sugar, it's a glucose plus a galactose, or maltose, which is malt sugar, which are 2 linked glucose units. So notice in the picture there is 2 monosaccharide subunits, and when that dehydration process occurs, that water is released. Polysaccharides are complex carbohydrates and can have thousands of sugar units. Polysaccharides have different structures and therefore names depending on where they are stored. In plants, this stored energy is called starch, while in humans, or animals and humans included, any unused sugar is stored as something called glycogen. Cellulose and chitin are structural polysaccharides, and so cellulose is the one that's found in plants, while chitin is one that's found in arthropods like insects, scorpions, crabs, and they can use that chitin to help build their exoskeleton. Lipids, also known as fats, are hydrophobic, meaning they do not mix well with water. They serve a variety of important functions in living organisms as phospholipids, steroids, and waxes. Stored fat can provide insulation or energy for later. They are also important in hormone production, as well as a very important part of membranes of cells, which we'll talk much more about cells later. A fat molecule known as a triglyceride is formed by the joining of a glycerol and 3 fatty acids, hence triglyceride. Note in the image, one glycerol is bonding to the 3 fatty acids. The hydrogen and hydroxides, or the OHs, will be released as water molecules as visible to the far right in the image. The fatty acids in a fat molecule can be saturated or unsaturated. What this means is that they have either all the hydrogen atoms that they can handle and are therefore saturated, or in the case of an unsaturated fatty acid, they are not completely filled with hydrogen atoms, but instead a different atom fills a spot or 2 or 3 and is then joined by a double bond. These molecules are considered not saturated by hydrogen or unsaturated fatty acids. The image shows fatty acids with side groups, which are indicated in red. In the fatty acid portion of the top example, every carbon is bonded to a hydrogen. This molecule is saturated. In the bottom example, there are 2 carbons that do not have hydrogens bonded to them, but instead the carbons have double bonded to each other. This is an example of an unsaturated fatty acid. The hydrogen atom's presence or absence dictates the properties of this lipid. Saturated fatty acids will be solid at room temperature, while unsaturated fatty acids will be liquid at room temperature. Think about it like this. If a fatty acid has all the hydrogens it can handle, it has more structure and can retain its shape at room temperature. If it is not full of some of the hydrogen atoms, it is lacking that structure and is therefore a liquid at room temperature. Unsaturated fatty acids can have one double bond or multiple double bonds and are named accordingly. Unsaturated fatty acids can be manipulated in a lab to increase hydrogen atoms or even change the conformation, the order of how the atoms bonded, which can have a negative impact on the health of people eating these foods. Phospholipids are fatty acids which are attached to a phosphate group. These lipids are essential in cell membrane functions, and we will talk more about these a great deal when we talk about the cell. As can be seen in the top image, a phospholipid has a hydrophilic or water-loving head and a tail of 2 fatty acids which are hydrophobic or water-fearing. These phospholipids form a barrier for a cell. 2 layers of phospholipids will align with heads out toward the watery environment and tails in away from the watery environment. You can see this in the bottom image of a cell membrane. Steroids and waxes have different structure from the rest of the lipids, but are also hydrophobic, meaning they do not like water. Steroids are important in the body as they help with cholesterol, which is necessary to synthesize many hormones in the body, and waxes are used mostly as a surface lubricant. Lanolin in sheep and waxy leaf coatings in some plants are examples. Our third macromolecule is proteins. These are made up of amino acids. Proteins serve many functions in living organisms such as transport, storage, and membrane properties. Enzymes, which help accelerate chemical processes, are proteins, and many hormones in the body, if not a steroid, are proteins. We'll talk more about enzymes later when we get deeper into the cell material. For proteins to function properly, they need a particular environment. If something is changed in that environment, the structure of the protein can be changed to the point it no longer functions properly. This is known as denaturation. Amino acids are the smaller unit or monomer which make up the protein. All amino acids have the same basic structure as noted in the top box on the image. They all have an amino group, a carboxyl group, and what's called an R group. Every amino acid has a slightly different R group, which gives that amino acid a function different from other amino acids. Notice the difference between the alanine and the valine R groups. These different molecules change how the protein will function in the body. Amino acids are held together by covalent bonds known as peptide bonds. A polypeptide is a long strand of amino acids bonded together. A protein is one or more of those polypeptide chains that has a distinct function and shape. So as I just mentioned, a protein is at least one polypeptide or more that has a particular function and a very specific shape. And there are 4 steps to completing this protein's final shape. So therefore, once it has a shape, it can perform its function. The amino acid sequence, which is how the amino acids line up, determine the primary structure. Next, this strand of amino acids will bend and hydrogen bonds will form, holding them into a secondary structure with bends and folds. The third step involves the interaction of the R groups attached to the many varied amino acids in the primary structure. What happens is this results in a 3D structure known as the tertiary structure. The final step is the interaction between different strands of polypeptides. Once these interact, the protein is in its quaternary or 4th structure and can then perform very specific functions, unlike all other proteins. Nucleic acids are the macromolecule responsible for life continuing. It is the basis of genetics. There are 2 types of nucleic acids in living organisms, DNA and RNA. DNA, or deoxyribonucleic acid, is found in all living organisms, from the simplest, tiniest single-celled bacterium to the largest, most complex organisms on Earth. DNA is a double-stranded nucleic acid, as in the image. RNA, or ribonucleic acid, is necessary for protein synthesis, like what we just talked about, which is important in the cellular functions. And as a comparison, RNA is only single-stranded. The base unit of nucleic acids are called nucleotides. Each nucleotide is composed of 3 main components: a 5-carbon sugar, a nitrogenous base, and a phosphate group. We will discuss nucleic acids in much more detail later in the sessions when we talk about DNA in Chapter 9. So that's the end of Chapter 2. Don't forget to review your end of chapter review questions, and if you have any questions beyond that, please email me.